everyone, and welcome to the Virtual Triangle SciTech Expo, sponsored by Biogen Foundation and the North Carolina Science Festival. My name is Hugo, and I'm going to be your host. So while we are waiting, I have a question for you. So this morning, we had a special program about the SpaceX launching. So were you able to see it with your naked eye? So write your answer on the chat. So we had here this special program and we were able to see it. And actually our expert is part of the astronomy days. So I'm pretty sure that he saw it too. So for those ones who are in Zoom, I'm going to go to share my screen with you for a quick Zoom tutorial, just to improve your view. If you're on Zoom, on YouTube, don't worry that we will take care of that. First of all, if you need closed captions, click on the CC button and set it up in the way that you need it. And to take advantage of the presentation and don't have like blind spots, so go to your upper left and change into speaker mode or speaker view, and then on the top, click on side by side. Between the image of our presenter and the presentation, you have a bar that you can move side to side just to make bigger or smaller each one. And of course, we want to know from you. So if you have any questions for our presenter, please write it down on the chat. And at the end of his presentation, we are going to ask him. And I'm going to stop sharing because it's time to introduce our speaker for today for this program, The Molecular Machinery of Life. So as I said before, if you attended our astronomy days, I'm sure that you already know him. Ron Monti is an outreach speaker for the Raleigh Astronomy Club and a volunteer educator and exhibitor for the North Carolina Museum of Natural Sciences. He is also a science instructor and volunteer for the OLLI Lifelong Learning Program at the North Carolina State University. Good afternoon, Ron. How are you?
I'm fine, Hugo. How about you?
Fine. So I can wait to see what you have prepared for us today. So please take it away.
Okay, thank you. I'm going to go ahead and share my screen. And okay, can everybody see that?
Yes, perfect.
Okay. Today's presentation, my presentation is called The Molecular Machinery of Life. And I'm going to be covering 4 different aspects of the machinery of the DNA molecule: the structure and scale, coiling and looping, replication, and editing and control. I have some perspective to share before we start, and that's that most of the general public is not aware of the astonishing complexity, intricacy, and sheer beauty of the molecular machinery within our cells. And I hope that you'll be able to see some of that during our presentation today. And we're just going to show one aspect of how truly amazing our cells are. So what is DNA? DNA is nucleic acid, which along with proteins, carbohydrates, and lipids is one of the 4 basic groups of organic molecules. And it's the information-bearing molecule which codes for the creation of proteins. Now, I'll just give you a little primer on the molecular structure of DNA. Here's the familiar double helix that you can see going up and down here. And it's kind of like a twisted extension ladder. So the side rails of the ladder in blue here are the sugar phosphate backbone of the DNA molecule. And the rungs of the ladder are the base pairs. And the base pairs are 4 nucleotides that form the genetic code. So when you see A, it's adenine, C is cytosine, G, guanine, and T is thymine. And there's A only bonds with T and C only bonds with G. So there's 4 possible rungs: AT, TA, CG, and GC. So here you see the ladder as if it was flattened out. You can see all four of those different possibilities down here. You can see adenine here joining with thymine, thymine with adenine, cytosine with guanine, and guanine with cytosine. So now the two sides of the rungs here, where the two base pairs, where the base pairs meet, those are joined by what they call hydrogen bonds. Those hydrogen bonds are fairly weak. They're not like a covalent bond where the atoms share the electrons. These are more of electromagnetic attraction for one another. So the hydrogen atom, which is positively charged here, is attracted to the negatively charged nitrogen atom on the left. And this weakness is actually a big benefit for the DNA molecule because they allow it to split down the middle when the cell needs to replicate. And it will form, each side of the DNA molecule when it replicates will form the complementary side of that ladder. So it will split and then it will form into two complete sections of the DNA molecule. And we're going to see that in the next slides. So it's important to understand scale. Cells in DNA are very small. And how small are they? Well, if this represented one of your human hairs, then you could fit 4, maybe 5 skin cells across the diameter of that human hair. Then if you enlarge that a little bit more, you'll see that this is one cell, and in the center is the nucleus. And in the nucleus are the chromosomes. This is a pair of chromosomes here, and we have 23 pairs of chromosomes in all our cells, except for red blood cells. So now I have one piece of homework for you, and that's the next time that you comb your hair, take out a strand of hair and hold it up to the light and see just how small that strand of hair is. I mean, when you see it on the screen here, it looks really big, but when you're holding it up to the light, you're imagining that there are 4 skin cells going across that diameter, and that one nucleus would be about 1/12 of the diameter of that human hair. It's amazing. So let's look at the dimensions of the DNA molecule using an analogy. And let's say that if you wanted to make the DNA molecule large enough to see, we'd have to make it something like the width of a human hair. So when you do that, the DNA molecule is only 2 nanometers wide. And to make it as thick as a human hair, which is 80 micrometers wide, you'd have to enlarge it by a factor of 40,000, which is a lot. Now, each of our cells contains about 6 feet of DNA. That's actually what's in our cells. All of our cells except red blood cells contain 6 feet. So if you enlarge that 6 feet to be thick enough to see, like the thickness of a human hair, that 6 feet would become 45 miles long. So that's the same as from Raleigh all the way over to Wilson, North Carolina. That would take about, it's 45 miles. It would take about 50 minutes to drive at 60 miles an hour. So just imagine driving that for 50 minutes alongside one long strand of hair. That's really amazing. And even just as amazing, there are 3.2 billion base pairs in the human genome. And even at the expanded size, there would still be 1,000 base pairs in every inch of that 45 miles. And that's too small to see. So now here's a cool illustration. I think it really helps illustrate what we're trying to express here. And this woman has about 2 feet of hair. Length of the hair is about 2 feet. And since a human has about over 100,000 hair follicles on their head, she has about 45 miles of hair on her head. So that would be equivalent to what we were just talking about, 45 miles of hair in one place. And also, if you enlarge the nucleus of a cell by 40,000, it would be about the size of a soccer ball. So now that amount of hair would be crammed into something the size of a soccer ball in the scale model that we're constructing here. So how do you do that without getting it all tangled up? I mean, sometimes I have trouble tying my own shoes, and yet our cells are able to take all this DNA, put it into something that size, and still be able to access any of the 20,000 genes that are in there, or to replicate that entire amount of DNA when the cell is ready to divide. That's a huge undertaking to do something like that. It's very complex. So the cell does it by coiling and supercoiling. And what is that? So here in this diagram, you see at the top, going across the top, the DNA helix. And this is kind of a coil in itself. So that's the first coiling. Then it goes down and it wraps around these histone molecules. These are 8 protein molecules, structures. There's 8 proteins in there that have to assemble before the DNA wraps around it. So that's another coiling there. Then those histones then themselves coil up into a structure known as chromatin. And this chromatin is how DNA exists in our cells most of the time. It's not in the form of the chromosome down here, the pair of chromosomes that you're familiar with. It's just halfway through the coiling process. So then after the chromatin is formed, it forms these loops. And they're known as loop domains, and we're going to see how those operate. And then it coils one last time to make the pair of chromosomes here. So you have a 5-level supercoiling here. 1, 2, 3, 4, and then 5 into the final chromosome. And as I said, most of the time the DNA exists in the form of chromatin. It only forms chromosomes when the cell is ready to divide. So one of the levels of the coiling is called histones. And this is an up-close view of the histone molecule. You can see it in the center here. It's these 4 colors here. So that's looking at it from one side. You see the 4 colors here. If you flip it over, you see another 4 colors on the other side. Those are the 8 protein molecules that form histone. And one of them has an extension here that you see down in the lower left. And that's kind of a control mechanism that will be showing in the slide up ahead.
Is the collection of DNA,
now, this is
protein.
Excuse me, I'm going to mute this. On the left, you see the chromatin. You can see like 1 histone here, 2, 3, and 4. And on the right, you see it uncoiled into a single strand of DNA. And that DNA in itself, both of those levels have to be unraveled before the DNA can be copied or replicated. So here you just see how it coils into a chromosome. But in a second, we'll see, here's the histones. And you see this molecule here is a chaperone molecule, and it's bringing an acetyl group to attach to the histone, that extension that we saw of the histone molecule. This is a chaperone protein that's bringing an acetyl group to attach to that extension. And that acetyl group will cause the histone molecule to relax and loosen its grip on the DNA molecule so that it can be copied. So there you can see the acetyl group here, it's been attached. And now you'll see it unraveling. And then there's also a methyl group. Well, here you see the DNA being replicated by this large molecule on the top. So now by unwinding it, let that molecule copy the DNA. And this is a methyl group that's attaching or a chaperone attaches a methyl group to the histone, and that methyl group can either tighten or relax the grip of the histone on the DNA. Now, this is a cool illustration. It's a virtual reality view of a nucleosome. That's the nucleosome is the DNA wrapped around the histone. And this is a tool that biologists use to get a better idea of what's going on in the DNA molecule. And you see this hand here. The person who's using this puts his hands into a glove, and they can, by using that glove and goggles, they can reach into the molecule and turn it around and expand it and contract it and measure it. So we're going to see that right now.
Series, we did talk about nucleosomes quite a bit, the way that DNA wrap around histones, as we can see here. And it's just great to be able to visualize this a little bit more accurately. So we can clearly see the DNA with the backbone in green, and then the proteins, we see all those alpha helices. So I and J here are going to be our two complementary DNA strands. So there's one, and then there's the other. And we can even highlight individual nucleotides here. We can see those being highlighted. And as we scroll through, it's important to see that there are 146 nucleotides or base pairs total, right? If we go 146 and 292, so that's 146 base pairs of DNA in this nucleosome. And then, as we said, these are going to be wrapped around these histones. And so there are 8 histone proteins, and we can highlight them one by one. So here's the second one. And so there are 4 different proteins, but there are 2 copies of each. So that makes this octameric disk. And we can go ahead and look at these one at a time. All of these proteins do have 3 alpha helices. So those are the red ribbons that we're seeing. And so all of the histone proteins do have very similar structure, though there are slight differences, like this one has this section projecting outwards. But on the whole, they're very similar, and they form this octameric disk. We can take a look at each one continuing here. And that is essentially the structure of the octameric disk of histone proteins. So once again,
yeah, that was a pretty cool tool. And I'll have a list of credits at the end of the presentation where you can go find this presentation online and see more about it. So that was at the histone and chromatin level of coiling. But just above that is the loop domain, which is another amazing way that these DNA molecule coils up. And a lot of people don't know about it. But inside this is, let's say, the nucleus right here. And each of these different shadings of the chromatin here that you see inside are chromosome territories. And the way that this is controlled for is by these loop domains. And we're going to see just how that works in the next section. And here again is where that loop domain exists. It's above the chromatin level, but before it falls into the complete pair of chromosomes.
In a recent study, we show that when the 2-meter-long human genome folds inside the microscopic nucleus of a cell, it forms 10,000 loops. Each of these loops connects 2 anchor points which are far apart along the chromosome, but which loop back, coming close together as the genome folds. In our new study, we find that loops form by a process called extrusion. Let's take a look at how this operates on a segment of DNA. First, an extrusion complex containing 2 subunits attaches to DNA, forming a small loop in the process. Next, the 2 subunits slide along DNA in opposite directions, making the loop grow bigger and bigger. Our simulations also showed that when loops form by extrusion, they lead naturally to the formation of what we call loop domains. This means that if 2 pieces of the genome are inside the same loop, they tend to bump into each other more often. In this physical simulation, you can see how the extrusion of 2 loops leads to the formation of 2 spatially segregated domains.
Yeah, watch how the red and blue stay in their own areas.
Like the convergent rule, the association between loops and domains was observed in our earlier study, but at the time we did not understand why this was the case. Both findings, and many others that you can read about in our paper, can be explained by the extrusion mechanism. Extrusion is an exciting model for several reasons. First, loops that are formed by extrusion will be unknotted, so if you need access to the genetic information they contain, it's no problem. You can just stretch them out. Second, extrusion doesn't just lead to segregation of domains within chromosomes. A collection of chromosomes that are all jumbled up will naturally become segregated if extrusion takes place. The fact that chromosomes are segregated in this way inside the nucleus, forming chromosome territories, is well known. Similarly, when several chromosomes are close together, loop formation by extrusion won't lead to entanglements. In contrast, loops forming by diffusion would routinely lead to entanglements. This is important because when cells divide, their chromosomes must come apart.
So you saw he called them anchor loops or loop anchors in the video. Those are the rings that the DNA, the chromatin, extends through. And this is a ribbon diagram of one of those loop anchors. And each of these ribbons here that you see are a string of amino acids to make the protein. And there could be thousands of amino acids in this string. And it's one string extended through here, through each of these different parts of the molecule. And they have to be assembled in just a specific way so that that protein folds the right way. And it's important because the shape of a protein determines its function. Now here you see, this is that loop anchor in the black and white ribbons you see here. And in order to function properly so that it can open and close around the chromatin, it needs an array of about 26 or 27 different proteins to help it do that. And each of these proteins have similar ribbon diagrams. So this is a very complex function here. And it's the cell has all the information it needs to do this on its own. Nobody's going in there and helping it do this. So it's kind of amazing. Now here we see loop domains. And this is a cross-section of a full chromosome. So like if you took the chromosome and just cut it across this section right here, you would see this. And each of these loop domains, you see it has an associated set of rings with it. And the inner set is red rings and the outer set is blue. So each of these loop domains have one red inner loop or inner ring and two blue outer rings to control that loop domain. Here's another view. Here you see the red ring and the two blue rings. So the red ring creates one loop and the blue rings create subloops within that original loop. Here's the cross-section of the chromosome again. And if you just looked at the red inner loops, or rings rather, you'll see that it forms a spiral going down. And here's where the loop domains in the different colors exist. And then down here, you can see the full chromosome. And here's a sequential view of how that is constructed here. So you see the red inner rings here, and you have the blue rings attached on top of it. And you start adding in the loop domains. And then when it's fully constructed, it looks like this. So those loop domains are quite complex. And when you add it together with the complexity of the chromatin and the histones, it's a major work of cellular engineering. So the next step we're going to look at is DNA replication and division. So here we have a pair of chromosomes inside a cell, inside the nucleus of a cell. And before the cell can divide to reproduce, the cells need to divide because cells are dying all the time. We have to make new cells to replace them. So before that can happen, the DNA has to replicate. So here you can see in this first step, the DNA is replicated, and then later in the process, those duplicated chromosomes split apart so that in the daughter cells here, each of the daughter cells has the same exact chromosomes and DNA as the parent cell over on the left. So this is what we're going to be looking at next, this replication process. And very roughly 60 billion cells replicate in our body each day. So now just to refresh what we talked about before, this is the DNA molecule. What we're going to see in the next few slides is how that DNA molecule splits down through those hydrogen bonds and splits apart. And each side of this DNA ladder is going to replicate so that we wind up with two ladders. Okay, now, what you're going to see in the next video is you're going to see DNA feeding in from the right. And this cluster of molecules here is called a repliposome. And it splits apart the DNA into two parts so that one half of the DNA molecule goes up here, and one half goes down to the bottom. And then these molecules here will construct the other half of that DNA ladder here and up here.
When DNA replicates, the enzyme helicase separates the DNA strands. Single-stranded DNA binding proteins then stabilize the unwound template DNA, keeping it in an extended single-stranded state so it can be copied by DNA polymerase. One new DNA strand is called the leading strand, which forms from its 5' to its 3' end by the action of DNA polymerase III. A sliding clamp forms a ring around the DNA and maintains the association of the DNA and the polymerase, allowing the uninterrupted synthesis of many thousands of nucleotides of DNA. The machinery at the replication fork includes helicase and a primase enzyme. Primase creates short RNA segments called primers on one of the template DNA strands. The RNA primers allow DNA polymerase to initiate DNA synthesis along the strand, forming short segments called Okazaki fragments that collectively form the lagging strand. A structure called the clamp-loading protein associates with a DNA polymerase molecule on both of the parental strands of DNA. The clamp-loading protein is also bound to helicase. These associations made by the clamp-loading protein coordinate the simultaneous synthesis of both DNA strands at the fork. Note that the two strands of a double helix run in opposite directions. They are anti-parallel. While the leading strand can be continuously synthesized in the direction of the replication fork movement, the lagging strand template must be folded so that its polymerase can create DNA in the 5' to 3' direction and still move in the same direction as the overall movement of the fork. Using the 3' end of a primer, DNA polymerase III lays down new DNA in the 5' to 3' direction until it reaches the previously synthesized Okazaki fragment. The DNA polymerase then dissociates from the DNA but remains attached to the clamp-loading protein. The clamp-loading protein adds a new clamp and places the DNA polymerase at the next primer. The process repeats.
So that was really amazing. And the next video you're going to see is more of a realistic animation of what's going on. But instead of the DNA feeding in from the right, it's going to be feeding in from the left.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion.
Yeah, that is amazing. When I first saw that video, my jaw just dropped. And to think that they left out a lot of mechanisms to avoid confusion only makes it more so. So here, I'm going to show you some of the slides that we saw, or this part of that video in more detail. So here you have the replication fork, the Repliosome. It's splitting apart the DNA that's feeding in from the right to go up to replicate each half, coming up to the top and down to the bottom. And then you see two DNA polymerases here. One of these drops off as soon as the next one is placed on here. At the same time that the helicase opens up the DNA and splits it apart right here, at the other end of the gap, another helicase molecule is splitting it open from the other end. So at both ends of this gap in the DNA, you have these replisomes moving out from one another. And thousands of these forks will work at the same time in any cell that's replicating its DNA. So along that 6 feet of DNA in the cell that's replicating, you're going to have thousands of these forks going on at the same time. So as it's moving apart, you see this one move to the right and then this move to the left, and it keeps getting larger and larger until it meets another pair of replisomes working in the other direction. And again, this is not showing the unwrapping of the histones. The DNA has to be uncoiled from the loop domains, and it has to be uncoiled from the chromatin and from the histones before this can happen like this. When you saw the DNA being copied in a certain direction, you know, they said they're anti-parallel from the 5' to the 3' section. It's talking about the sugar phosphate backbone of the side rungs of the DNA. And because they're anti-parallel, that's the way the chemistry works. That's how atoms work. The way that life evolved, it didn't choose to do it in this complex fashion. It had to do that in that fashion because the way that molecules are, the electromagnetic characteristics of how molecules form. So now here we see the helicase and the RNA primer. And then it talked about these binding proteins in the first video. These binding proteins keep the single strand of DNA open so that it doesn't loop back and fold on itself. You know, if it's like if you had a piece of Scotch tape and it folded back on itself, you can't use it anymore easily anyway, because it's stuck to itself. The same thing is happening here. If you didn't have these binding proteins, the DNA might fold back on itself and you couldn't replicate it. Here's how the replication fork is the flow of the DNA is working. This is the DNA polymerase here, these oval shapes. This is the molecule that actually duplicates the second rail of the DNA molecule here and here. And then you have the DNA clamps. The function of this is to hold that RNA polymerase onto the DNA while it's replicating. If this clamp wasn't here, the polymerase would fall off and it would dramatically slow down the replication process. And this is the clamp loader, which is an amazing molecule. And in the first video, you saw how the clamp loader loaded on the DNA polymerase, and it also loads on the clamp itself. And this is kind of how it works. This is the clamp loader, and the structures you see on the right, these three forks here, hold on to the DNA polymerase and attach it to the single strand of DNA. And then the other part of it takes the clamp and then loads that onto the strand of DNA right next to the DNA polymerase here. And here's how it works. You have, in this natural state, the clamp loader is closed at the left side. Then a molecule of ATP, which is the energy-bearing molecule that's used throughout the body, attaches to it. It opens the end of the clamp loader. The clamp loader comes along. The clamp loader opens the clamp and attaches it over the strand of DNA and lets it go. And then the clamp closes. And this is a ribbon diagram of the clamp. You can see, like the other molecule that we saw, that it's quite a complex molecule. And this has to be constructed from the genetic code to perform its function. And here again, I'm just showing how the DNA has to be unwrapped from around the histones. And just the wrapping and unwrapping is a complex process. Here you're seeing the DNA feeding in from the left. It has to be uncoiled from around the histones here. And then after it's duplicated, the histones have to be recombined with the DNA. You see all these molecules here that have to be used to help it recombine around the histones. And then there's two other things we'll discuss briefly, is the DNA editing, which is going on here, and the unknotting. Here's that other diagram we saw a second ago. How much DNA is in our body? This is amazing. The amount of DNA in our body, in all of the trillions, there's 37 trillion cells in our body. About 12 trillion contain nuclei. And they contain, if you attached all the 6 feet of DNA in each one of them together, it would reach from the Earth to the sun, well over that distance. And the amount of DNA our body produces in a lifetime is more than a light year, which is 5.9 trillion miles. So here's our solar system. Our solar system is embedded in the Kuiper belt. The Kuiper belt is embedded in the Oort cloud. And the Oort cloud is like several light years across. Our bodies produce close to that diameter of DNA in our lifetime. Let's think about that. How accurate is DNA replication? After all the processes are complete, it's accurate to 1 error in 10 billion pairs. That would be the equivalent of typing 60 words per minute continuously for 38 years with just 1 typo. But that's day and night without stopping. So Snoopy did a great job there. Still, with 3.2 billion base pairs per cell, that's roughly equivalent to 1 mistake per cell every 3 days. Another aspect of what we should look at is how the cells need to keep from tangling, the DNA from tangling. Now, you remember what we were talking about here. Well, there are mechanisms to do that. And here's a video to show you that.
Is that even though we often think of it as a code or a logic problem or something something like that. When it comes down to it, it's also just a gigantic amount of string inside the very tiny nucleus of a very tiny cell. There are a couple of types of enzymes inside the nucleus whose only job is to run around untangling DNA all day. If two lengths of DNA are crossed, one of these enzymes can hold one, break the other, let the first pass through, and then relink the part that it broke. If a length of DNA is being wound too tight from the strands being pulled apart for replication, then another one of these enzymes can come, break one of the strands, let it untwist, and then stick it back together. And that's how your DNA doesn't become a huge knot. This is Sophia Tintori from the Dunn Lab at...
Okay, so here's one of the molecules that unwinds when the DNA gets wound too tight, it breaks one strand here. And this is the one that breaks one strand of DNA and lets another pass through. So the first strand of DNA, it would rest across this molecule here, this protein. And this protein looks like an anchor. It would rest in these arms here, going horizontally across. The arms would grasp it, break it apart. And the second strand would come in through the top, go into this opening here. After the second one is broken, it lets it pass through, then joins the second DNA molecule going across, and then releases the first. And this is just a brief sketch of what it was doing there. First one comes in, breaks the second down here, lets the first one pass through, and then joins the second one back together again. So here you have, this is a different mechanism. If DNA gets broken by something like UV ray damage, there's some mechanisms that they have to repair it. And one is down in the lower left to repair a single rail break. And this other molecule on the right is to repair a double strand break, which is very complex to do. You can see each of these little circles in this molecule is an atom. So here's let's skip that. This shows you what happens if there's a double strand break. So say the top molecule, this is a strand of DNA. This is from, say, the mother, and the father's DNA is down here. If you get a double break, this molecule can't use the other strand to repair itself because it doesn't have the other side of it to tell it what to put together again. But there's this mechanism, amazing mechanism that's going on here. So now, say radiation comes in and breaks, causes a double strand break. See it break through? It's not just causing this break here. Other damage occurs right in that same area. So it has to have these other molecules come in and take out all of the damage there. So now, how does it repair itself? It doesn't have the opposite side to repair. The father's DNA opens, the mother's DNA comes in and repairs itself. Now, one thing I should explain is that we, in those pairs of chromosomes, it's 23 pairs, one of those pair is the DNA from your mother and one is from your father. And they're all duplicate genes, so that if something happens to one, the other is used to replace it. And that's one of the ways that it's being used here. So now this other molecule comes in, reconstructs the mother's DNA by copying the father's. Then it goes back, you see it go back. And now it can copy this part up here. Now it has the information it needs to construct the other side of the rung here and over here as well. So there it did it. And here it's doing it for the other side. And then finally, another molecule comes in and glues where the break occurs together. So now, I'm just going to skip through this slide because we're running short of time. And there are many, many other very complex processes going on within our cells that we would be truly amazed to see. And I hope that you got from this presentation just how complex DNA is. And if you want, you can take a print screen of this screen, and it shows the credits for where those videos came from. And you can go on to, these are all from YouTube. You can go in there and just key in a search for these terms here, and you can find those. So that was the end of the presentation. If you have any questions, I'd be happy to try and answer them.
Sure. So we have some questions from the chat. So Adam wants to know, when the hair grows, are your hair cells dividing?
No, they're not dividing. They're producing, I believe, proteins. So that's occurs during protein synthesis, that those cells can create new proteins which form the hair itself. Plus, keratin, I believe, is the primary protein in the hair, and those are proteins which are produced by the cells.
Mm-hmm. And he wants to know too, because the chromosome shapes are really cool. So what factors cause DNA to take up these cool shapes?
The molecules that form the structure of the DNA coil and supercoil, they are proteins that the cell has created over the course of evolution. And I'm not totally familiar with the molecular mechanisms that cause those proteins to form in the first place, how the cell determines to make those proteins. There's a very complex set of cell signals. It's called cell signaling, where the cell knows which proteins to make. And those proteins, when they're formed, those proteins are each a very long string of amino acids. And because of the electromagnetic charge and polarity of those amino acids, when they join together, it causes those proteins to fold in a certain way. And from there on, it's just a matter of the cells following the electromagnetic forces, combining those proteins to coil that cell up. And when you think about it, it really is amazing because, it's doing it without somebody there to tell it. It's doing without a computer program that's being edited to make it do that. It's just doing that on its own because of the electromagnetic charges on these cells, causing it to coil and fold in different ways. And the way that those cells find where to go is by the action of the molecules inside the cell. It's what is the term for that Einstein had or the motion of cells, the motion of atoms or molecules inside. It's a Brownian motion. It's something like Brownian motion that's causing the cell, these molecules inside the cell, keep bumping around one after another, and finally they run into the one that they need to combine with. And it happens very, very quickly. And you would think that it would not be an efficient way to do it, but actually it is, because it happens so quickly.
Mm-hmm. You were talking about when the DNA replicates that we need the half from one part and the other half from the other part. How can you explain these species of animals that they can clone themselves without the need of a mate?
That would be something that would be covered under reproduction. When I don't know exactly how they go about cloning a cell. I do know that in what we talked about in this presentation, The cell gets these signals from either itself or the outside cell that it's time to go into the replication cycle, and it divides at that point in time. As far as these other life forms, I'm assuming that it would do the same thing when it needs to divide to replicate itself. There's some signal coming from somewhere inside the cell, creating a transcription factor that latches onto the DNA that causes it to start the replication process. Something like the helicase that you saw in the in this video here.
Mm-hmm. And as the last question that I have for you is that we have seen you in astronomy days talking about planets, space. Now that you're here talking about all this DNA, so it's like a different world. It is a different field. What is your path? So what took you to this point that you are interested in all these fields? And what is your inspiration?
Yeah, my career was not in science, it was in accounting. And when I retired, I had this long time interest in science, and it gave me a chance to study more about it. And, I took things like, have the great courses, how they have these courses that you can study on biology and astronomy. And I joined the astronomy club, and I volunteered at the museum there. And I began picking up more and more about what was going on that we never really see, that the general public never really sees. And I like to think that whereas maybe some scientists delve too much into detail so that the public can't understand that kind of complexity, that I might have more of a general public understanding of what you might need to see to kind of understand what's really going on there, for the general public to understand. And I enjoy talking about it. So the museum gives me a great chance to do that.
And it is amazing just to hear all the things that all the process and the things that we have inside of our body. That is so big, and we know so less. Like what you were saying, just to compare the sizes with the space, it is amazing.
Yeah.
So I want to say that it's time to wrap up. So thank you very much, Ron, for being with us.
My pleasure.
And thank you, everybody, for joining us for this talk. And thank you to our sponsors, Biogen Foundation and the NC Science Festival. And of course, thank you for all members of Friends of the Museum, because without them, we couldn't do programs like these ones. So we have another program for today, and then you have all day tomorrow. So tomorrow we are going to have robots. We're going to play with Legos. We're going to know what happened with the Perseverance, and we're going to have our famous guest and sketch. And the thing is going to be Animal Kingdom. So Miranda, Kerry, and myself, we're going to be there doing our best to draw things and just play with the animals. So again, check out those programs and more on our website at naturalsciences.org. And if you missed any of our programs or you want to rewatch this one, we are going to post it on our YouTube channel. And we have an escape room, a virtual escape room on our website too. So if you think that you're able to escape the genomic lab, just go to the website and play and let us know if you could able to escape. So Ron, thank you again. And thank you everybody, and see you in another program. Happy Friday. Bye.
Bye.